All right, perfect. I think we have a good enough people to at least get started. My name is Brenton Pyle. I work at DNA Nexus, putting on thought leadership events like this. I'll be giving a brief introduction and just running through some housekeeping things before I turn it over the floor to our experts to just begin the panel discussion. So thank you again for joining us today for our massive scale image analysis roundtable. This will be recorded, as you should see when you join. This will include the Q&A section at the end. So if you have any hesitancy around having your question recorded, it will only be read aloud via text from our panelists. But if you have any hesitancy around that, you can always drop off now and catch the recording later. Recording should be available within about 24 hours. It'll be available on our YouTube, and we'll send it out via email. How to get your questions answered. There's a Q&A box that you'll see in your screen. That'll be the best way to get it to us. We'll be reviewing it throughout the webinar and answering questions at the end. And if you don't have any of your questions answered by the end, you can reach out to us at community@dnanexus.com. Now I'd like to introduce our panelists for today. We have Aaron Chu from Flywheel. We also have Carl Land here and Joe Herman from Regeneron Genetics Center, and our moderator, Ben Busby, who will be leading the discussion. So Ben, I will pass it over to you.
Awesome. Thanks, Brenton. So I am particularly excited to be in this webinar because I am a genomics person by training, but really over the last 2 or 3 years, I've really started to be able to integrate image analysis and really image-derived phenotypes into my analysis. And I'd really like to give a big shout out to Oliver Gray, who's in the audience, as well as Felix and Stephen Smith from Oxford for helping out with some of those things, and Andre Klempier, who was unable to be here today. I'm really excited to be with this panel. I think almost all of you are familiar, probably everyone in the audience, with the Regeneron Genetics Center. Many of you are probably not familiar with the fact that they do a lot of image analysis, and they're going to tell you about that today, as well as genomics integration. And I'm also very excited to have my friend Aaron Chu here, who knows lots about biomedical data of many, many forms, and currently works at Flywheel, which is really a place specialized in large-scale image processing for both experts and non-experts. I'd like to start with Joe and Carl. Joe and Carl, could you tell us? I'm interested in what you personally work on, but first, could you tell us a little bit about the Regeneron Genetics Center and really what it's doing in imaging these days?
Sure, yeah, thanks, Ben. Thanks for the intro. As you see, Regeneron Genetics Center has been involved in the exome sequencing for Biobank as part of the large consortium that's been spearheading that. And so we have had access to all the large-scale exome sequencing data, as well as sequencing data from other cohorts in-house. And what we're really interested in is leveraging that in conjunction with deep phenotyping data that comes from a variety of different sources. So that might be electronic health records, but also increasingly sort of multimodal data sets that come from different omics technologies. And then on top of that, kind of a newer thing, which has really been enabled Biobank imaging project, is putting on imaging phenotypes into the mix as well. So really what we're interested in is finding genetic associations with all these imaging-derived phenotypes, which increase our power to discover associations and so in some cases help us to really better understand the disease. In our team, we're especially interested in leveraging some of the newer imaging data that's coming in from Biobank to enable discovery as part of our discovery efforts. So largely, I guess we can do like a little intro of our backgrounds, and Carl and I, and sort of maybe talk a little bit more about the specifics of some of the research that we're doing. My background is in statistics, genetics, and bioinformatics. I've worked in a little bit in biotech, and then got very interested in sort of these large-scale problems. How can you really start to move from small research projects to really large scale projects where you can leverage the information that's contained within hundreds of thousands of genomes. And particularly, that's really exciting when it comes to rare variant associations, which you can only see when you get to this type of scale. So Regeneron has access to a lot of these different data sets, as I said, and so that's how I came into this role. Maybe, Carl, you can give a little bit about your background.
Sure, yeah. So my background is originally in physics, and then I did a PhD and postdoc in medical resonance physics, so MRI and actually kind of other subtypes of MR, where you can look at non-water species and measure those. And I joined Regeneron about 3 years ago. I originally joined in the clinical imaging team, where we have ongoing clinical studies, and you want to analyze the imaging data. And I was working on that, and I worked a little bit on the UK Biobank with the RGC, and then I kind of realized that it's really cool. It's a unique opportunity, right? I mean, I come from my background, MRI physics. Like if you look at the papers in the fields that we publish, typical sample size, I'm not making this up, in these journals like MRM, JMRI, things like that, is like 6, right? I wrote one paper with a sample size of 1, okay? So, when I saw that people are now analyzing imaging data sets when we were doing it 3 years ago, it was like 40,000. Now we're up to 70,000, right? These are like totally different questions that you can now answer. The tools that you need to use are totally different because computational considerations become huge when you're working on 70,000 instead of 1. So I thought it was just this super unique opportunity, and I really pushed actually to make the switch to join the imaging team within the RGC. And about 6 months ago, I did join. So yeah, that's pretty much, I guess if you want to hear what we're working on now, I mean, it is large scale image analysis, mostly UK Biobank, but not just UK Biobank, right? I mean, anywhere where there's an intersection of genetics and imaging, that's things that we're focused on. And I don't want to get into specific project details too much. A lot of it's unpublished, but, broadly things we do is something like, if there's some Git repo or Docker or something that analyzes some, let's say a brain MRI or something like that, and we're interested in these image-derived phenotypes, IDPs, I'll be the guy who implements that. If there is no tool out there that does that, you actually have to develop it from scratch. Maybe there's a paper written, maybe there's nothing at all. We come up with novel analyses, run it at scale. We also work on that. So a little plug, I mean, if people have in the audience kind of algorithms, packages, pipelines, things like that they're interested in using on UKB, and maybe they don't have computational resources or expertise or something like that to run it on scale, please reach out to us. We're really interested in all diseases, all imaging types in the UK Biobank. So we're always looking for collaborations. So yeah, please reach out. Ben, I think you're muted.
Hey, thanks, Carl. And also, I'd like to note, I have some follow-up questions for you and Joe, but I want to get to Erin first to let her introduce herself. But before that, I would also like to just give a little bit of a shout out to the UK Biobank community. So UK Biobank now has a community page. If there's things you want to talk about, you can jump on there. Questions about imaging, of course, would be welcome, and discussions about imaging would be welcome. So that's even a broader place you can discuss. That said, thank you, Brenton. And the link is in the chat, by the way. And now I'd like to go to Erin. Erin, I'm actually personally curious. What really drove you to move full on into the imaging space? And then later I'm going to ask everybody about imaging plus genomics. But let's talk about imaging, just imaging for now. What drove you to move into that space fully?
Yeah. And so I'll be very honest here. Brenton told us what questions you're going to ask us, Ben. So I don't want to give away too many of the answers as to why I moved over to imaging, because that'll spoil my other responses. But just give me a little bit of a background. So Ben and me go way back because I used to operate in life sciences data distribution strategy and management at Amazon Web Services. So that is where my background is kind of understanding how do you manage big data? Well, cloud is a great way to solve a lot of the problems when it comes to storage, management, staging of compute, finding compute resources. And because of the sheer volume of genomics data in my own background, which is a PhD in molecular genomics as well as a veterinary doctorate, genomics data was where I spent a lot of my time. Now, when we started to move into imaging, some of the biggest projects that I helped to work on before I came to Flywheel were things like the Imaging Data Commons, which did a huge scale of harmonizing openly accessible cancer imaging data and making those data readily available into cloud service providers. I understood that the imaging space was just a totally different frontier. And so the reason I came over to Flywheel, Ben, was because I saw the opportunity to become an expert in a space where I've been peripherally involved because multimodality is the direction everyone is going in. And Flywheel is the expert and the platform for medical imaging data management. What Flywheel does is end-to-end data management. So that is everywhere from getting the data in, indexing it and cataloging it. I will spend hours talking about indexing and cataloging biomedical data. You know this, Ben. Flywheel does it better than I've seen because we take advantage of some of the inherent attributes of medical imaging data, specifically the DICOM data format. We can help to stage and deploy workflows automatically on data in place. And we go all the way to, can you go ahead and start developing machine learning algorithms? We provide for that within the Flywheel platform. And can we help our customers collaborate with other customers through data distribution and exchange? So we really are end to end, and it's been a real ride for the past, wow, almost 4 months now with Flywheel. And so I'm excited to share what I have learned coming from that genomics big data space and applying that into imaging.
Oh, right. So two things. One, audience, you heard it here first. Multimodal data is the future, as stated by Aaron Chu. Second, Aaron, I will almost certainly go off the rails in terms of asking you guys questions. So I think prepping may have been a bit of a fool's errand, but that's okay. I want to go back to Joe for a second. Joe, I know you're personally involved. I know you're a statistician by training, but I know you're really personally involved in integrating sort of imaging and GWAS, FWAS data, those types of things. You mentioned rare variants earlier. Why is having massive amounts of image data so powerful in your hands?
Well, so yeah, I mean that's obviously the key question is what is imaging enabling us to do that we couldn't do with transcriptomics, other types of multi-omics data sets. Why is it a big deal? Why do we care so much about this? So I would say that, I mean, there's two angles you could take really. One is that a lot of the diseases that we're interested in studying, diagnosis is made via imaging. So if we can get an imaging-derived phenotype that is very closely representing what is used clinically as a diagnostic tool, that's obviously a lot more informative than a billing code in an EHR in terms of telling us about what's actually going on with that patient. So in that sense, it's a much more direct report on the status of the patient in a way which is actually relevant. So if we can find genetic associations that associate with imaging-derived phenotypes, that's potentially a more direct route to really understanding the underpinnings of the disease. And then I guess the kind of related but slightly orthogonal point is that often we've found, and I think this is seen in other places as well, that the imaging-derived phenotypes give us a lot more power to assess these things statistically. And part of the reason for that is we move from having a binary encoding, which, as I say, you'd have in the EHR, which is noisy in itself, but is also just often just a 0 or 1. To something which is continuous quantitative trait. So we can not only look at whether somebody has a disease or doesn't have a disease, we can look at the whole spectrum of severity levels in a quantitative way, which often gives us more statistical power. So we have found in a lot of analyses that actually associations which have sort of a low effect size when it comes to looking at the binary traits actually end up having a much larger effect size on the quantitative imaging derived traits. And that means ultimately that we can find signals with a smaller sample size and potentially find things that you couldn't even see from an EHR. So from a statistical perspective, there's definitely a lot of added power that we get from having these traits.
That's fantastic. And I totally have an off the rails question that I'm going to ask you in 2 minutes after I talk a little bit with Aaron and Carl about disease
Sure.
subtyping and image analysis, which I'm personally very excited about. But first, Aaron, so obviously you come from a genomics background, but you've moved into imaging. I mean, there's probably a lot of people like that in the audience, right? Who are really genomic scientists and moving into imaging. Any simple pieces of advice?
Yeah,
Folks.
absolutely. So, the first thing I would say to my genomicists in the community out there is please enjoy the metadata. Um, I will say that coming from the genomic space where we are literally scratching in PDFs and maybe some tables and publications looking for disease phenotypes and information on the age of the patient or this data, whether it's an initial or a secondary visit. I enjoy the DICOM metadata. So there are inherent DICOM headers that are embedded in the study itself. Those things can be a gold mine. So get comfortable with things like PyDICOM, where you can start to really look through those DICOM headers and understand that those headers are things that you can mine in a machine queryable fashion. So that is absolutely something I'd recommend, is just enjoy it, because I literally felt like it was Christmas when I started diving through these headers, that you get such rich information about the study. And as we all know, in the biomedical space, context is everything. The other thing is keep in mind that size is relative. A big study in medical imaging data can be a couple of gigs, and we are comfortable saying that big data in the genomic space are a couple hundred gigs, if not terabytes of data. But those data, the size will go insane without you thinking about it, because the sheer number of individuals who have had imaging studies compared to the folks who have had sequencing is so much greater. So even though a single study, when we talk about the difficulties transferring some of these data, yes, maybe one study will be a couple of gigs, right? Depending on the modality, it might go into dozens of gigs, but all of a sudden you're looking at terabytes or petabytes of data, simply because of the size of the archives a lot of these provider groups have amassed. So when you start to think about storage, think about it in terms of patients and as well as the longitudinality of the studies.
Awesome. So what you're saying here is that random VCF headers are just not sufficient. Never mind. I'm not even going to ask.
No.
That said, I mean, I think that's why sort of the biobank movement is going, right? I mean, like that's like biobanks have sort of changed the game for us in the genomic space in terms of having some phenotypes. And again, I'm going to ask Joe about this in a minute. That said, Carl, you obviously come from more of an imaging background than a genomics background. How was your entry into starting to sort of mix these things and really work? You work in one of the world's largest genetic centers, right? Like you're embedded in a milieu of genetics professionals and genomics professionals. How's that working out for you as an imaging scientist, and how does it work to integrate with them now?
Yeah, I mean, that's true. I come from a very different background than over 90%, probably 98% of people at the RGC. So, the language they speak in some ways is a lot different than what I'm used to, and vice versa, of course. I mean, I think for me, if you want to see the perspective of an imaging person going into genetics, some major tips would be, find a genetics person to work with because from my background, coming from physics, MRI, and things, you really have to understand kind of like a small number of concepts very deeply, and that's kind of what it is, right? In the genetics, I find the opposite. Like, there's this massive amount of information and jargon and things like that I'm not really used to, and none of it's conceptually necessarily that difficult, but you're just lost. So if you have somebody, I have Joe here, and then there's tons of other colleagues who can help kind of translate it. I think that really helps a lot. And then I would say, again, for an imaging person coming into genetics is, find a niche. Obviously, the niche is imaging, right? So I'm not going to be expected to be some, world expert on genetics of X or Y or something like that. But I should be probably one of the most competent people in image analysis and things like that. So I'll give like a very brief one. If I have give some advice for people coming from genomics into imaging, I think it really depends on what their background is, right? I mean, a big thing would be like, learn kind of classical image analysis. And I would define that as image analysis up to deep learning, right? And deep learning really changed things. Then the next part would be learn deep learning, right? But I mean, we really use both, honestly. So, you use both in nearly every project. So it's not like, deep learning totally removed the need to understand, classical image analysis. Deep learning is something obviously that takes a lot longer because it hasn't really been ingested and it's a continually changing field. That one's a lot harder. A single intro classical image analysis textbook will get you really far in that regards. And then, I'd say for large scale image analysis, learn Linux. I don't know, maybe not everybody knows it. Maybe most people do. It depends on your background. I learned Linux from coding on the console of an MRI scanner. The GE scanners run Linux. So if you wanted to automate reconstruction and things like that, you had to do it through Linux commands. So that's my only exposure. And it's funny, I spent 10 years learning MRI physics. I don't use a lot of it. I use Linux all the time. So that's like the most important skill that I picked up through 10 years of kind of, post-grad work, basically.
Totally understood. I mean, Aaron's dog is also very excited about Linux, and just a small plug too. If you are an imaging person coming into genomics, check out Bioinformatics and Functional Genomics by Jonathan Pevsner. I think it's an amazing introductory textbook to sort of to sort of mirror some of the things Carl was saying. We're going to ask Carl a little bit later about really strategies for getting image-derived phenotype pipelines in through GitHub, through Docker, those sorts of things. Personal plug for learning containerized strategies like Docker. We have a webinar on that we did with HDR UK. Queuing Brenton, thank you. And then also, so I'd like to go back to Joe, though, and talk a little bit about phenotype data for a second, which is a bit off script. But I guess, I mean, one thing that I think about all the time, and Aaron probably thinks about, or at least used to think about quite a bit too, is that ICD codes are really clusters of diseases, right? We all know that Alzheimer's disease isn't a disease, right? Like that's ludicrous, really. And so do you find like in the really real world that like images and image-derived phenotypes are helping you really sort of classify some types of diseases and make these, you mentioned earlier, these like SNP and multi-SNP associations tighter? Do you think that's because you're really optimizing for one subtype of disease?
Yeah, I mean, that's a very good question. I guess there's sort of a research answer to that and then a kind of practical answer, maybe. So first on the sort of practical side, ICD codes may not represent some kind of real construct biologically, but they do represent a real construct when it comes to the clinical world. And that's not just for billing, that's also, in terms of treatments and approvals of drugs and so forth. So we do have to work with those as real entities and to some extent. And that's something we always keep in mind with our imaging analyses, is that at the end of the day, we want to know that whatever we're finding can be translated back to some clinically meaningful entity, whatever that is, whether it's ICD code or other ways of defining phenotypes, Phe codes. There's a few different ways people use. So that's sort of like the practical angle. On the other hand, from the research angle, yes, I mean, I think it's clear. Certainly in some diseases, you have the hierarchy within the ICD-10 system. You have subtypes and so forth. But even within one code, or subcode, you might have actually clusters of diseases or a spectrum. And one of the things that we're excited about is whether we can unravel that, disentangle the different subtypes, or sort of better classify them based on distinct imaging features. And some of that's already, out there. There's, as I said, there are some diseases that are diagnosed via imaging, and the subtypes of diseases like macular degeneration based on what the retinal phenotypes look like from an imaging perspective. Those would not be treated the same clinically. They might receive the same overall ICD code at a high level. So we do distinguish between those when we're analyzing them, and the imaging is obviously critical for that.
Awesome. Thank you. And I don't know, spoiler alert, people, we're going to talk about deep learning at the end, and I want to come back and talk about sort of if not deep learning, some learning, some on that. But first, I want to ask a few other questions. And Aaron, this is my first example going off the rails. If we could play off what Joe said, but think about longitudinal data, right? He mentioned that, I mean, I talked about sort of subtyping, but he mentioned sort of a stage of disease, right? And can you talk a little bit about how massive scale imaging analysis helps folks define stage of diseases and how that plays into phenotype?
Yeah, I mean, staging from a clinical perspective speaks to how you are going to treat an individual. It's also going to speak to how you might give a prognosis for an individual. In our case, what we see a lot in our customers is that they are trying to ingest longitudinal data to track the status of the disease over time. That means the data gets a lot bigger. It also means that you need to be able to carry over annotations from one stage to another of a study. And so another thing that I will pass along here, that's the beautiful thing about germline DNA, right? Is that it's in theory, it shouldn't change. You sequence the individual once, you might sequence a tumor multiple times, but when it comes to imaging, you not only need to make sure that you are imaging the same individual, you have the annotations from the past set of images, but you also need to ask questions like, was the imaging protocol the same? Was the device the same? How are you getting all of those data together? And so one thing I will also point out, and Carl, I really appreciate the fact that you said to learn Linux. We recognize that not everybody is going to pick up Linux. I was forced to learn Linux at some point in my PhD, and it was a painful endeavor for me. And so Flywheel does try and serve those clinical researchers who may not have a computer science or a bioinformatics background. It is basically a browser-based platform where you can ingest data. You can actually index different longitudinal studies on the same subject and actually and view them and export annotations and relay them onto new studies. So when you start to think about longitudinal studies, think about the fact that you need to manage to carry over annotations and comments and analyses from one time stage to another. And that's going to be your big question too, is has the lesion changed? Has treatment been involved? Has the individual's prognosis been altered based on X, Y, or Z changes in your parameters? Not having all of those data in one place is going to be a huge hindrance if you are going into that longitudinal space.
So perhaps as a helpful tip for those of you using UK Biobank data on the rep, there's a concept of data set extenders. So you can take image-derived phenotypes from a platform such as those made from Stephen Smith's lab, sorry. And we did a webinar on that earlier. Put those in as additional fields that are tagged to patients. So, and if you were a DNA Nexus commercial customer, you could theoretically take data from Flywheel and put it in there. That said, I'd like to go to Carl. Carl, could you give people who are sort of data scientists, probably familiar with basic Linux, sort of tips on pulling image analysis software from GitHub and Docker really into sort of large scale cloud analysis on the UKB wrap as well as elsewhere?
Yeah, for sure. I mean, we do a lot of that. Probably half our analyses are essentially taking tools from the literature, from Git repos, from Dockers, things like that. Provided the license allows us, of course, but if it does, which is always nice, then the procedure is always pretty much the same as to how we actually run this at scale on all, we're 70,000 images we have available, depending on modality. Essentially, we build what's called an applet in DNA Nexus, where, this individual applet, let's say, you can launch a bunch of them at once as well, like a bunch of workers. let's say you have 70,000 images, right? And you want to run 1,000 workers. So then each individual worker will process 70 images, right? So there'll be some kind of loop. You can even parallelize within that. I won't get into that. But within the loop, essentially, it finds an image, that it wants to process. It downloads it locally, and then you run you either pull that package, like the Git repo or the Docker or whatever, and then that does something, right? Often they're using the Docker. It's like built through a command line interface, right? Where the input's kind of some kind of input image, you specify that, the output's the output image, and then you have to you have to move your data into the Docker to run that. Then you have to take your data out of the Docker, and then you upload that where it then lives for future analyses or in order to depending on what the package does, it might just process the image and kind of output that, or it might also produce a set of IDPs directly, right? Lots of these packages come with their own kind of analysis tools built in, which is also nice. So, yeah, we really appreciate when the kind of community at large makes this available for us. What we really don't appreciate is when people put up like dummy Git repos where it's very clear that it's totally unusable, and they put it up probably just to satisfy reviewer comments. But we spend time trying to implement these, so it's much better if it just doesn't exist, right? And I'd say it's about 50-50. I can usually tell. I've implemented probably 30 of these. I can tell, is this a real or a fake by looking at it in a couple minutes. But sometimes if we really want it, I'll spend even if I think it's fake. And sometimes you can get it working in the end. It's just like it takes weeks. So yeah, I don't know if you want any other further details on that.
No, that's okay. I think all of the folks in genomics have also felt this pain, so we can stop there and not relive our nightmares.
I would just also add in terms of reducing pain, something that we use a lot of, and which has definitely streamlined a lot of our analyses, is the DX whittle functionality. So that really helps with kind of going from n of 1 up to however many tens of thousands we might be looking at. So definitely appreciate all the work that's been put into that.
Hey, Joe, thanks for mentioning that. Yeah, I mean, I think workflow languages for everybody are super helpful. And I think heretofore they've been mostly used for genomics. But yeah, I mean, moving workflow languages into image processing and all that, I think makes a ton of sense to me. That said, Joe, this is something that's been rattling around in the back of my head. When you're doing sort of genomic and imaging analysis together and you're looking for associations, how do you know when you have enough genomic sites for an association, right? Like, I mean, we all know that like when we do a GWAS, we're typically not just looking for one site, multiple sites interplayed, so on and so forth. How do you grab that like sort of sufficiency to say that this image, like explains a lot about sort of maybe an etiology, a genetic etiology, and this genetic etiology really explains what's going on in that image?
Yeah, so I mean, maybe one way to address that question is sort of coming back to the question of rare variant analysis, because I guess that's sort of part of what you're asking is, how do you have enough samples who carry these different genetic variants to be able to see some differences within the images. Obviously, if your starting point is a very noisy imaging-derived phenotype, you're going to struggle to find some very subtle genetic effect on that phenotype. Actually, we find in a lot of cases that that's not the case, that we actually find very heritable DPs and we're able to pick up a lot of signal. And actually, surprisingly, even when the raw phenotype looks very noisy, often that noise is non-genetic and doesn't really impact the analysis. So we are able to find a lot of signal, but where the sample size question really comes into play is if you're dealing with rare variants, and then obviously, if you've got something that's 1 in 1,000, 1 in 10,000 even, the number of images that we're going to have access to is going to be quite small from carriers of those rare variants. So we have a bunch of statistical methods that have been developed both in-house by others for kind of boosting the power of some of these rare variant analyses. And that definitely helps with, sort of aggregating signal across different variants and being able to see, for example, on a kind of gene level, what the impact of variation in that gene is on the imaging-derived traits. I think there's a statistical genetics aspect of that, and there's also just sort of an image processing aspect to it. Sometimes we can squeeze more power by doing a better imaging analysis, by filtering out low-quality images, etc. Sometimes we really need to just go back to the kind of methods from the genetics side.
Thanks. Yeah, no, and I have a question for Aaron and then a question for Carl and Aaron, kind of following up directly from that. So Aaron, I guess the first thing I would ask you is, so you made a bold claim, which I think is yet to be seen, and also working in the biobank space, we see sort of more of a balance, even a little bit more on the genomic side. But you made a claim that, right in sort of real world evidence, there's way more imaging data than genomic data, which is obviously true. So how do you flip that on its head? How do you deal with it when you have way more imaging than genomic data, and you're trying to figure out sort of genomic phenotype associations through imaging? I'm wondering sort of how you think through that problem.
Yeah, I mean, so I will say that the majority of Flywheel's customers are just edging into that the intersection of genomic and imaging. Ultimately, though, it's exactly what Joe said. It's really dependent on what you're looking for. So I'll give you an example. If you're looking at a rare disease or pediatric disease, there's no way you're going to get statistically powerful enough data samples just within your institution. And so what we're seeing a lot is just cross institution and global consortia that are trying to up their sample sizes by saying, we are looking specifically for this disease and we're looking for these attributes within this individual's imaging, or vice versa. We need to find these variants within this individual's genome. So that's one thing, is collaboration. But the fact is, right, if you're looking for an imaging phenotype that is driven, that you think is associated with a genetic variant, then you're limited by your smaller sample size, which is going to be the sequence, right? It's going to be the genome. And so I will say that we do have folks also who are saying, okay, we think that this imaging phenotype is correlated to X, Y, or Z genetic variant, and we're going to try and sequence these individuals. It's hard, right? Because then you have to assume that the original study was going to allow for recontact of the participant, plus or minus the individual complying to give sequence data. I honestly think start with the genetic data, and you're right, there's a plethora of imaging data out there. You can try and correlate. After you've found an initial correlation, go ahead and look at imaging studies that don't have a correlated genetic variant. But, maybe I'm biased here, but from a genomics perspective, you're always going to be limited by the number of genomic sequencing samples that you have.
So in terms of reconsent, though, I'm taking my prerogative to argue with Aaron, because we've known each other a long time. Our future health is coming, right? I mean, 5 million theoretically reconsentable people. Like, this is going to break this mold. Like, you're right. We've been stuck in that paradigm for the last 20 years. But I mean, maybe this is the first biobank that's going to change things. I don't know. But
I'm looking forward to it.
Absolutely. I want to rocket off one more thing that Joe said, but for both Aaron and Carl, but I'd like to ask you to keep your answers short, because I want to get to machine learning and then make sure we have time for audience questions. And my question here is a little bit complex, but he was talking about how, I mean, if you're, and I'm doing some inferring here, but he was talking about images and thinking about things that are related to the phenotype of interest, and then also peripheral things that are not related to the phenotype of interest. And obviously, if you're looking at abdominal MRI, people do all kinds of things that damage their organs for all kinds of reasons, but they may not be related to genetic etiology. And so I guess the question for Carl and Aaron is, how do you think about teasing apart parts of images and image analysis that is related to a phenotype of interest, and parts that are actually effectively noise? And how do you clean that out when you're thinking about image-derived phenotypes?
Yeah, I think that's a good question. I think it's a very complicated question. I think it depends a lot on the specific question that you're trying to ask and answer. So Yeah. So, I mean, I'll give, I guess, a concrete one. From our perspective, right, if you're looking at abdominal MRI and you're interested in finding some, genetic traits that affect something of interest, um, or some genes that affect some trait of interest, if all these other things are uncorrelated with the genetics, then when you do the GWAS, that just ends up being noise, right? So we might not even be super interested in it in that perspective, right? And a lot of what we're doing, in the end of the day, we run these IDPs through GWAS and XWAS. So from that, I'll give that from the genetics perspective. Now, from the imaging perspective, it might matter a lot, right? And what I'll say is, like, if you're building a model that tries to, let's say segment, which is something, probably 80% of our pipelines end up using, abdominal MRI, maybe you're interested in the liver, you have to segment the liver before you want to run that analysis, right? If you have all these wonky livers that are totally damaged in, all these different ways, you obviously have to put that in your training data, right? And you do have to, go to kind of great lengths to span the diversity of, normal physiology, disease physiology, imaging quality, all these different things in your training data. Otherwise, when you go and run it across the 70K, your model's going to be garbage, right? If you just train on healthy people with nice livers, see what happens. And we've done this. It's not even just gonna fail in ways you would expect. It's gonna fail in, like, laughable ways. Basically, the models don't really work like we do. So yeah.
No, and
Wow, that was
yeah, no, go
powerful.
on, Ben.
No, go ahead, Aaron.
No,
It's you.
I was going to agree with Carl. I mean, from a GWAS perspective, that's exactly right. If you're seeing a bunch of not even comorbid, but um, co-occurring phenotypes, that's going to wash out if you are actually looking at a real signal. But I was going to give an example. So we ran a body part classification model the other day, and it was trained on a bunch of just radiographs of the thorax, and the appendicular skeleton. And we were basically saying, hey, we were asking how well can we classify this body part on radiograph. And I was actually spot checking the results of the model because it's performing pretty well for things like arms and legs. And then I was looking at hips, and we actually had one that was like, this is definitely not a hip. It's like it had like a 40 or 30% confidence that it was actually a hip on the radiograph. And I opened up the image, and it was actually an individual who'd had a total hip replacement, right? So your data, your model's only as good as the data you train it on. And so we had never trained this particular, the training set did not have individuals that had rods in their hips. Right. So that's exactly right. If you know that there are comorbidities associated with the phenotype you're looking to track, you need to make sure that your training set does actually include individuals that have those comorbidities. Otherwise, they're going to be caught as noise.
That's amazing advice. Thank both of you for really throwing that out there. That's great, and I think it segues perfectly into sort of our last segment before we get to audience questions, which is, I mean, thinking about applying learning algorithms, and obviously our friends and colleagues are throwing learning algorithms at all kinds of questions in all kinds of ways, and obviously there's very powerful new algorithms out there. I've always, in my sort of personal leveraging of and with colleagues, obviously, of machine learning, I've always found it much easier if I was sort of doing an association where I had a binary outcome. But obviously in this space, we don't have so many binary outcomes. On the one side, there's sort of longitudinal progression and these other sort of noise characteristics we were talking about. And on the other side, we have these sort of multiple etiological groups with multiple mixtures and so on and so forth. So any advice for really leveraging, I guess, so I guess what that means is we're really looking at deep learning approaches, comparing lots of things to lots of things. But obviously you can have all kinds of, all the way from just totally erroneous results to hallucinations, so on and so forth. How do you guard against that? How do you validate after you have sort of deep learning model associations? And that's for everybody.
I mean, I'll take a stab at just sort of running off that, but I guess in some cases, what we're using deep learning for is, as Carl said, segmentation of regions of images. So in in those instances, being slightly wrong, slightly getting a slightly different boundary of a region um, may not actually make any difference for a large-scale genetics analysis. So, I mean, in some senses, those types of uh, questions and phenotypes are a little bit more robust to errors versus something where you're trying to kind of do a binary classification of, is this feature present in an image? Is it not there? So I think in some cases, we have found that continuous traits are a little bit more robust to errors in that respect, or sort of biases maybe in the model versus something which is forced to classify things as either binary or even multiple classes. So yeah, that's maybe sort of one area where I'd say um, it helps you if you're looking at something which is a continuum.
No, I think that's really pragmatic advice, right? Like run faulty models on fault tolerant problems. Uh,
Yep, I'll have some kind of musings about this as well. I mean, again, I think it depends on the specific question. If you're, say, like, let's say building a classifier that predicts Alzheimer's from a brain, right? And you could probably do this. And like you said, well, Alzheimer's is really a whole bunch of other diseases, right? So at that point, I think you're too late, right? You've already set up the question that you're, solving for Alzheimer's instead of Alzheimer, like the set of Alzheimer diseases. And the model will learn, okay, there's various different types of Alzheimer's, but, whatever kind of various, basically subregion sizes or things like that, atrophy in various regions, but that's all Alzheimer's. It is just going to learn that. It's not going to have a problem with that, right? It doesn't know. So I feel like at that point, it's too late. And so, one thing that people do is, they like to cluster these days, and people are always hunting for ways to cluster subtypes of diseases in hopes that you can treat them different and they're biologically different. And I mean, so I'll give an example from the literature of the things, people do is, let's say you, instead of doing that, you build an autoencoder that essentially blocks out parts of the brain and then reconstructs the brain from those missing portions. And so essentially, it's like, the input is the brain with the patch missing. The output is as close as the original brain as it can possibly make. And it's not going to do it perfectly, but it's going to do, a pretty good job. And if you look at the central layer where it's kind of compressed most, they call that an embedding. And then you cluster that embedding. It's just a set of values. You might find clusters, which is to say, while some people, have these kind of, how you map those clusters back to something interpretable, that's a whole nother problem. And most people just kind of sweep that under the rug and they say, who cares? The point is, what if we GWAS them? Oh, they're genetic, therefore they're real, right? That's kind of what people seems to do. But you could look at that, and at least you would see, are there different clusters of disease in your imaging? And if there is, maybe that's an indication that you're looking at multiple different diseases. Now, you notice I talked about brain MRI. I didn't say anywhere that those clusters necessarily will be subtypes of Alzheimer's. Could be subtypes of any disease, right? So again, interpreting it is very difficult, but I think, people kind of look at that and they'll look at, tries to way to cluster that, and then maybe those different groups will kind of respond to, or kind of longitudinally will develop differently. And then that's kind of an indication they hope that this is kind of a real clustering and not just a hallucination of the clustering algorithm or the autoencoder, all these other issues. So it's all kind of wishy-washy, but, it's a difficult problem. So, and yeah.
So Aaron, if you want to weigh in before we get to audience questions, that's great. Or the first audience question is actually for you. So either way.
Oh, sure. Well, okay. I do want to weigh in just because Joe and Carl make a really good point, which is you may very well be solving for several outcomes, right? Depending on what your algo has picked up. If you are saying, like, I'll give you an example, right? If you are literally saying, if you are telling your machine learning algorithm, I want you to identify a lesion on this abdominal, CT. the first thing you're going to need to do is clean your data. And I think I'm going to say, like, we're all data people here. That's the first step, is always to clean your data. And that's hard, coming from the fact that imaging data can be super heterogeneous based on who operated the machine and what the machine was and when it was taken. And so the more you can harmonize the way you pre-process your data and try and denoise, get rid of motion artifacts, right? There are open source algorithms that are out there. Make sure that you have a staging pipeline that has your data being pulled in and cleaned to the best of its ability. That's going to just be one step to just make sure you're not accidentally running an association on a motion artifact. And that's something that we think a lot about at Flywheel, is how can we enable our customers to not have to think about that pre-processing QC and QA. We were talking about Docker containers, Ben. We actually work to Dockerize algorithms that we know are used in the imaging community and make them literally point and shoot on Flywheel itself. You can actually stage them so that when data hits the platform, it immediately will deploy a bunch of what we call gear rules, which are just bunch of pre-processing algorithms that'll have the data in a derived form ready for you to use. But that's the first thing I'd say, is like, it doesn't matter what environment you're working in with, like, have a robust QC and QA pipeline, and make sure you feel as good as you can about the data coming in before, worrying about what is it actually answering? What question are you actually answering?
Gotcha. So I'm going to ask a final Jeopardy question. I'll tell you what it is now. Please don't answer this question yet. This is for everybody. But is hanging your data on some sort of knowledge graph, either pre-hoc or post-hoc or both, after you feed it into a deep learning model, going to be useful? But before we get to that question, Aaron, are there specific models your team is using for image analysis? We know there are probably hundreds, but are there multiple options that you use to prioritize the tools, other than accuracy, precision scores, or other parameters?
Yeah, so I will also clarify, Flywheel, we do have a team of scientific engineers who are doing science. Typically, it's to optimize tooling for our customers who are working on the platform. So we're not running our own de novo research the way the folks at RGC are. And so it's been a real pleasure to hear what you guys are up to. But the way we prioritize what tools we are going to develop, as well as what, like specifically, whether it's an algorithm or just something as simple as, a pre-processing QC/QA tool, is going to be based on demand. And then performance is secondary, because the fact is, if you see like a tool like, I'll give you an example, Presidio or something. We know that that tool, which is pixel recognition and pixel scrubbing of PHI, it was actually trained on a very small number of sample sets. You can fix that algorithm, right? You can look at it, you can retrain it, you can improve upon it. But we knew that there was a demand from the community to have that functionality. So that's what drives us, is first demand, second, we'll make it better if we can.
Cool. All right. We have about 30 seconds for each person to answer the final Jeopardy question. Let's start with Joe. Joe, knowledge graphs. In, out, deep learning, both or neither?
Well, so I guess knowledge graph is one of those phrases that's kind of come and gone in different incarnations over time. I don't know if it's sort of, I think, taken a bit of a sideline with all the talk about large language models. People, I think, have this idea that you can kind of bypass the actual structure and just sort of get a model to figure out how everything's related. But yeah, I think, I mean, to the extent that you can build tools to help organize the data that gets and the conclusions that get generated within an organization and make that reusable, especially, I would say, that, I mean, this is something that's probably worth mentioning for everyone who's involved in tooling. A large part of what ends up being done as part of training some of these models is manual annotation by experts, and that may be us, but it may also be, domain experts in, especially with the imaging, with somebody who knows a lot about those images. So being able to reuse a lot of that expert knowledge across different problems, ways of storing that, ways of managing that, linking it back to the imaging features, that all that type of metadata and sort of knowledge in general would be super useful.
Well said.
Thinking about, but that's something we're thinking about.
That's helpful. Carl, 1 minute.
Yeah, I'll need less than that. I've never heard of a knowledge graph, so I don't know. I'm gonna punt on that one.
All right. Well, we will leave you to Dr. Google or GPT,
Yeah.
whatever your flavor of choice is now. Aaron, last word on knowledge graphs
Yeah,
and so
yeah.
on.
I think you're right. I think knowledge graphs kind of had a beautiful spotlight, and then we're just kind of unceremoniously pushed off stage by some of the advances we've seen in LLMs in the past few years. But I see no harm, and I honestly think that it would be great if we did have something more holistic where we'd be ingesting structured metadata, annotations, information about a subject, and be able to map that across the across projects. I will say we haven't done it. But I think that there is value in it, especially for groups that are tracking huge amounts of data over time.
Awesome. With that, if anybody's going to BioIT World, there might be a session on knowledge graphs and large language models.
Oh,
Uh, but anyway, I would like to really thank our panelists. Thank you, Aaron. Thank you, Joe. Thank you, Carl. I think this was really an eye-opening experience for a lot of the people here. Thank you to the audience for staying. And as always, thank you to Brenton for organizing and putting all this together. I hope you guys have lots of sort of questions coming from this, and you have the opportunity to reach out to all of us in the future if you need to. Thank you very much, and have a great day.